Hello everyone, and a very warm welcome to this year's PacBio Workshop as part of the Plant Animal Genome Conference. My name is Jonas Korlach, I'm the Chief Scientific Officer, and it's my pleasure and privilege to host and moderate today's event. It's so thrilling to see all of you and that we are able to meet in person again, even though I didn't imagine I would be wading through a half foot of water this morning getting out of my hotel room. We have our theme this year is Actionable Genomics at Scale, and we have a terrific program for you covering all the different areas of plant and animal genomics. We'll talk about agrigenomics with Gina's talk. Brett Schaefer is going to talk about biodiversity and conservation genomics. Sarah Carey about evolution and functional insights, and then Dan Portick will talk about metagenomics. But before I give the stage to the speakers, I would like to make a few comments about the new systems that we announced. So I think you all know that we announced two new sequencing systems, one in long-read sequencing and one in short-read sequencing. And our goal here was to really take and optimize and scale the different technology to where it is best suited for the different applications, whether it is long-read sequencing or whether it is short-read sequencing, because using the best suited technology in each application clearly gives you the best results. So starting with the Revio system, this system has a 15 times higher throughput than the Sequel IIe sequencing system, and that's accomplished by interrogating 100 million reaction vessels compared to 8 million on the Sequel IIe. We reduced the acquisition time to 24 hours from 30 hours, and so what that results in is a system that can give you 360 gigabases of highly accurate long reads of hi-fi reads in a 24-hour time period. Going into that in a little bit more detail, we've designed the system from the ground up to deliver hi-fi at scale. And so the scale I talked about, 25 million ZMW smart cells, and you can run 4 of them independently, simultaneously in a 24-hour cycle time. But we've also optimized and improved on all other areas of a sequencing system. With regard to the compute power, there's 20 times the relative compute power in the Revio system compared to the Sequel IIe system, which is largely based on GPU architecture. For a little bit more for the geeks among you, there's a program called Deep Consensus that we developed in partnership with Google, which optimizes the circular consensus hi-fi calculation. Previously, you had to do that offline, which was computationally expensive. On the Revio system, it is now on board, so you get deep consensus called hi-fi reads right out of the box. With regard to the ease of use, there's a 50% reduction in the consumables. There's a load in advance capability, which lets you load the next run and prep the next run while the previous run is going. And there's a wide time window at which you can do this so that you can really maximize the time that the system is running. And there's no longer a nitrogen gas requirement like you had on the Sequel IIe, so no longer do you need the hookups and all the plumbing for the nitrogen. With regard to the cost, the US list price of a Revio smart cell is $995. It yields about 90 gigabases of data, so about 30x coverage for 3 1 gigabase genome size genomes. And so at the scale of a 1 gigabase genome size, that's about $330 for a reference quality genome. It takes under 1 minute to load the instrument, and while the amount of data has tripled from 30 gigabases on Sequel IIe to 90 gigabases per smart cell on the Revio system, the data output has only increased by 1.5-fold because we did some optimizations in how we encode the information and how the data is being stored. So then, as I mentioned, you have 4 independent stages that enable flexible experimental designs. So just by example here, on cell 1, you can have a maze sample. Pooling, indexing, and barcoding, of course, is fully enabled like it was already on the Sequel IIe system. So on the second smart cell, there's a mixture of oak and ladybug. Then we have mistletoe, and then we have a completely different application, MOS-seq, that I'll tell you about in a minute, on the 4th smart cell. And so the overall annual throughput then on the system, again measured against 1 gigabase genome sizes at 30x coverage, through these improvements is about 4,000 genomes per year, a dramatic improvement, as I said, 15 times improvement compared to 250 genomes per year on the Sequel IIe. So we wanted, at the time that we announced the system at the ASHG conference about 2.5 months ago, we showed and also made available to the community some human data sets. We did the variant calling and benchmarking and confirming that Revio provides accurate data. For this conference, we wanted to highlight, of course, some plant animal data. So in collaboration with the researchers at the Sanger Institute as part of the Darwin Tree of Life project, we pooled 2 animals and 2 plants. So you have, they have species that they use as control systems to testing new methods, new protocols, technologies. And so ladybug and oak are some of those. And so you can see here in this table, the ladybug and the mouse gave over 90 gigabases of data, 91.2 gigabases of data. To give you a sense of the quality, over 90% of the data is Q30 or greater, 99.9% accurate long reads or greater. And then we pooled them in such a way with the known genome sizes to account for getting enough coverage for the de novo assembly. So for the ladybug, which has a smaller genome, we get excellent assemblies. There are 2 numbers for the assembly sizes with regard to the size and the contiguity, because as you know, with hi-fi sequencing, we get fully haplotype resolved assemblies, diploid assemblies. So you get 2 genome equivalents per sample. That represents the maternal and the paternal, the biological truth that you have 2 copies of a genome in each biological cell. Now for the mouse, that's different because that's a highly inbred strain that's been used by the research community, and so that's why there's only 1 number. And then the second run was 2 plants, an oak and a mistletoe. You see, again, the specs for the oak assembly are excellent. Contig N50 is well over 10 megabases. And so for the mistletoe, it says not applicable because that genome is enormous. It is actually 30 times the size of a human genome. And you may have seen that just before Christmas, in time for the holidays, thematically appropriate, the researchers from the Darwin Tree of Life project announced that they had completed the assembly of the mistletoe genome, 100 gigabase size genome, 30 times the human genome. In fact, you can see down on the bottom right, that's drawn to scale. That's the size of the human genome. It's 10 chromosomes. So there are chromosome arms in mistletoe that are larger than the entire human genome. It's just truly remarkable. And so congratulations to the Darwin Tree of Life team for this tremendous achievement. We were trying to help raise awareness. We have a blog post series that we call the Hi-Fi Difference. And so in our Christmas edition, we were commenting on this. If you're not familiar, go to pacb.com/blog. And you can also subscribe to get regular updates. Going to the agrogenomics space, we collaborated with our next speaker, Gina from Corteva AgriScience, to sequence maize. And she's going to talk a lot more about that. I'm not going to steal much of her thunder. This particular SMRT Cell was a little bit lower with 70 gigabases. And I wanted to mention this because we're not cherry picking data. This is how things come off the instrument. Still plenty enough to give enough coverage and a really excellent assembly. This assembly is in 59 pieces. And considering that maize has 10 chromosomes, that's less than 6 pieces per chromosome, straight out of the hi-fi data for the assembly. Now from the Kmer plot on the right, and on the left you see the size distribution was about 15 kilobases. And look at the y-axis, that's the accuracy. So well above Q30, the mean is Q36, Q40, so almost 99.99% accurate reads coming off the Revio system, and that's because of the deep consensus improvements that I mentioned. Now on the right, on the Kmer plot, you can see that this is also very homozygous and inbred. So what about much more heterozygous and also polyploid plants? We collaborated with Jeremy Schmutz from Hudson Alpha and Scott Brainerd from the Savannah Institute to tackle the hexaploid persimmons. So now you have 6 haplotypes of about 800 megabases. So overall, that's bigger than the human genome as well. We ran 3 Revio SMRT cells. We wanted to have enough coverage. Again, you can see 270 gigabases divided by 3 is about 90 gigabases. And then this Kmer plot on the left, I think, is really pretty because you can just count the 6 peaks that correspond to the 6 haplotypes. We have about 57x coverage for each haplotype. And this assembly is in progress, but Jeremy told me that the assembly is really excellent with a contig N50 of 12 megabases for both the maternal and the paternal, for which each you have 3 haplotypes. And so just to give you a sense of the quality of the data and what the data set looks like, what we did on the right was a quick hi-fi ASM assembly. Then we took the longest contig and mapped 1 smart cell's worth of data back to that contig. And so in unique regions, you get the read pileup, and then IGV has a function that automatically phases and groups the haplotypes. And so you clearly see the 6 different haplotypes. The one on the top is fully concordant with the contig, which is what you want to see. And then the quality of the data, the phasing of the different variants across these regions, and how easy it is then to distinguish, and then you can do your biology. Now, the last example of a genome that I want to show you is in the area of pangenomes, which there are many talks here, so I'm going to be very brief because, of course, just sequencing one individual doesn't give you the full complement of the genetic content of that species. And so pangenomes, to recognize the biodiversity within a clade of sequencing multiple individuals, either from the same species or from a different taxonomic, gives you insights about the core genes that tell you about the biology, the critical functions, and what makes that species. But then the dispensable genes are so critical for understanding the adaptive traits and what the potential of that clade is to adapt to climate change and so forth, because they can evolve much more rapidly. So in collaboration with Mike Schatz at Johns Hopkins University, we took 8 nightshade samples. We had 1 tomato as a control. The next one over is the wild progenitor of the eggplants that we're now eating. And then 6 lines of the Ethiopian eggplant, also known as pumpkin on a stick for the reason that you can see here in the picture. And so we pooled 4 samples together, ran 2 Revio smart cells per pool. So in a day's worth of sequencing, again, about 360 gigabases or 90 gigs per smart cell of data, and then running hi-fi ASM. I know this is a busy table. I just want to focus you on the 4th left column, which is the coverage. So we had plenty of coverage, well over 30 gigabase. These are 1 gig genomes, so well over 30x coverage, in some cases even 50x coverage. So we probably could have even taken 10 or maybe 12 samples pulled together in this Revio run. And then the column, the 4th from the right and the 3rd from the right, really showing excellent assemblies, 1 gigabase size. These are mostly inbred. Contig N50s, again, 40, 50 megabases Contig N50s. So remarkably, we've shared, so this was done relatively recently, about 2 weeks ago. We then shared the data with Mike and also his student, Katie Jenicke. And Katie has a talk tomorrow at 11:30 describing her new tool called Panogram, which is a tool to visualize and extract information from pangenomes. And so Katie and Mike gave me a preview of her talk yesterday, and I was really blown away. So if there's one talk that you want to see on pangenomes, go to Katie's talk tomorrow at 11:30. It's really a tremendous real-time tool that they were able to use to deduce already really important biological insights about these species. And this is just remarkable because just a little while ago, a project of this magnitude would have taken months to just generate the data, and then we didn't have the tools to really mine the pangenomes, and now within a span of 3 weeks, this is just accelerated so dramatically because we now have the tools and the sequencing machines to gather the data quickly and at scale, and that facilitates the tool development to really show the tremendous benefit that that brings. So check out Katie's talk at 11:30 in Pacific Ballroom A, and then Mike also is going to talk a little bit about it in the afternoon tomorrow as part of the Galaxy workshop in the Ballroom D. Now, of course, it's not just the sequencing that you have to scale, the entire ecosystem of upfront sample prep and data analysis. So earlier this week, we announced a new program we call PacBio Compatible, which partners with the industry, with the community to work on solutions on all different aspects. So for example, with Hamilton and TCAN, automated library prep that is giving you from the sample to the finished library. On the data analysis side, partnerships with DNA Nexus, with Google, I mentioned already, and so forth. So this is a program that will help scale and appropriately scale the entire throughput. So related to this, you have seen on your chairs is we have a passport program where several of these partners are listed, and also some of our customers. And so if you'd like, we'd encourage you with this passport program to visit them and learn how that interfaces with PacBio. And if you get the stamps, you can come by our booth and get a prize. Now, the second part of my talk, I'd like to spend on multi-omics capabilities of Revios, because now that you have your beautiful PacBio HiFi genome assembly, there are other things that you want to do with it. You want to annotate that genome. You may be interested in the epigenetics component, and you may want to understand the different functions of the different cell types by generating a cell atlas. So with regard to genome annotation, it's been shown now for many years that iso-seq, meaning full-length RNA sequencing with PacBio, gives substantially more complete information and also corrects errors that were made with the previous short-read technologies. So on the left, you see one of the earlier papers going back to 2017, where the chicken transcriptome was built with iso-seq, discovering over 20,000 novel transcripts, correcting over 4,000 erroneously merged events. So what happens here in RNA-seq, you have so much coverage, so many reads, that you get noisy alignments, and it becomes difficult for the software to understand the boundaries of the genes. And so on the bottom here, you see that in the genome expression atlas, there's one genome annotated, but the PacBio data actually show that it is 2 genes. And on the right, this is a more recent paper on ant brain annotation and transcriptome to understand the sociobiology of ants. You have the opposite problem. So in the previous annotation, there were 2 genes, whereas iso-seq, the reads very clearly show that it's just 1 gene. This is not a solved problem. Last year, there were papers on Arabidopsis, a new transcriptome atlas. Even in human, there are corrections that are being made. So we are certainly anticipating that with the 15-fold increased throughput on the Revio, which brings down the cost, increases the number of reads, this will become the standard for a more accurate and more comprehensive annotation. In addition, there are updates on data analysis with regard to variant calling and phasing and identifying allele-specific expression. So in 2020, we introduced isophase, which can do this, but only for SNPs and not in a sort of most user-friendly environment. Now the update, we have deep variant from Google that can call SNPs and indels in both exons and introns. There's some really clever transformations that keep the exons together and so forth, showing that with deep variant from Google, you get substantially better performance even at quite low fold coverage. Deep variant will be incorporated into the SmartLink environment for variant calling, so you can get this out of the PacBio environment. Now, with regard to methylation detection, I think you know that since April of last year, the Sequel IIe is a true 5-base sequencer and the only on-instrument 5-base sequencer on the market. So in addition to your 4 bases, you also get 5-methylcytosine straight out of the box, and the same is true for Revio. So we've looked at this on human data, which is the best case because of the ground truth samples. So on the left, you have your 4 bases, and then overlaid, you have the information color encoded with the methylation. So this was on human. We looked at animals and plants. This data was kindly provided by the Vertebrate Genomes Lab, where we looked at this toad, and I'm just going to show a couple of screenshots that all look, show that it's working. Basically, you have the gene in the middle there in this blue track, and you have the corresponding non-methylated regions that are transcriptionally active at the promoter, and there may be some enhancers and so forth. Here's a fish, the chub mackerel, the same kind of thing. So all of that looks normal. And even while I was browsing the genome, I found this region where you can clearly see indications of allele-specific methylation. So this region, which is grouped by and phased by haplotypes using the SNPs here, you can see that this region is highly methylated, whereas this region is unmethylated. So you get methylation in a haplotype resolved manner, just like you would the genome assembly as well. With regard to plants, again, Mike Schatz and his, at the time, postdoc, Shujun Ao, who's now got his own position at the Ohio State University, looked at the tool that we built called Primrose to, on the left, first confirm the performance of HiFi-based methylation calling comparing it to bisulfite sequencing, and you see there's a very nice relationship. And then on the right, an example again from nightshade plants showing that all the red arrows that you see there are gaps where you do not know the methylation status with short-read bisulfite sequencing, because these are repetitive elements. You don't have any reads that map, and so you don't know what the methylation status is. The top track is the Hi-Fi-based methylation that gives you contiguous information. We took a first look on Revio. This is, again, going back, this is one of the tomatoes that I was mentioning. And so this is a homozygous inbred line, so it's all homozygous. And you see the typical picture that you would expect, where you have regions that are unmethylated in blue that are transcriptionally active versus the rest of the genome that is methylated. Now, the term epigenetics is used both for methylation as well as chromatin architecture in dynamics. And so there's been some great work over the last 2 years by 2 groups independently at UW and at UCSF developing methods to adapt Hi-Fi sequencing to measure chromatin accessibility, open chromatin versus heterochromatin, with the paradigm of having highly accurate long reads. I don't have time to go into that in detail. So there are some preprints and papers. The left one was in Science 2 years ago. There's a tremendous new preprint looking at telomeres, looking at the chromatin architecture of telomeres. That's never been done before. We were not able to access this region because it's so repetitive, and with short reads, you just don't know where you are. And now look at this. You see this beautiful nucleosome footprints down here, the array spacing. You see the subtelomere being more highly accessible chromatin, and on the right, you see the transcription factor accessibility. So this is now accessible with the same kind of quality, contiguity, and comprehensiveness that the genome and the transcriptome is provided by PacBio sequencing. So lastly, single-cell sequencing and building a cell atlas to understand the different cell types and their cell functions. And of course, it's been known for a long time that alternative splicing is a very critical component of giving cells, and in some cases, the causal element for giving cells their particular function. And so what do we know about this process? For example, in humans, it's known that 95% of all human multi-exon genes are alternatively spliced and have more than one isoform. The average number of isoforms per gene is estimated to be 7 or greater. So just one example here, this is from the brain. This is just one gene called BIN1, and what you see in these colors is, and the legend on the right, are all the different isoforms of that one gene. And then on the x-axis, you see the different cell types in the brain, and you can see dramatic differences with regard to isoform usage. There was a recent preprint showing that 12%, about 2,900 genes that are expressed in the brain, are only heritable at the isoform level. What that means is that it matters when you have allele-specific expression, you have allele-specific splicing, and you need to understand the different isoforms. Different isoforms can have different functions from the same gene. Sometimes they can have the opposite biological function, different isoforms from the same gene. Now, you don't understand any of that biology by just counting 3' or 5' ends with short-read RNA-seq. You need to understand and resolve the full transcript, and this is enabled with single-cell iso-seq. We have now commercialized this into a kit, which we call MAS-seq, high-throughput, full-length single-cell sequencing. The method is adapted from research by the Broad Institute, and it is based on concatenating, in this particular case, 16. We're stitching together 16 full-length transcripts. And the rationale behind this is that the average transcript coming out of the 10x Genomics Chromium box is about 1.5 kb, but you know a Hi-Fi read can be 15 to 20 kb, so there's a lot of capacity there. And so by stitching together 16 of those molecules and then sequencing those, we can increase the throughput even further by 16-fold. This kit is available for both the Sequel IIe as well as the Revio. And what's been shown in the preprint is that with this throughput increase, you no longer need short reads to build your cell clusters to identify the cell types. You can use it all with one technology, but now giving you the full information about the isoforms. So here's an example, a recent example, where we've run this MAS-seq single-cell full-length sequencing on Revio. This is a human example, 9,000 cells, one Revio smart cell, again, 94 gigabases, 7.2 million reads, but now with the concatenation, that translates into over 100 million full-length transcript reads. and you can run 4 Revio smart cells simultaneously, so that's over 400 million full-length reads per day. So that's about a 200-fold increase in the capabilities compared to Sequel IIe without concatenation. And here's one example of why this is important, or how impactful this is. Now, this example is in the human space, actually cancer. But they're using this concatenation to show, number one, what I said, you don't need the short reads anymore to do the cell type clustering. And then they show that with this additional information, they were able to identify that there were events misclassified erroneously from the short read data. So short reads failed to distinguish these events, potentially leading to wrong biological conclusions. And the authors stated that long read sequencing provided a much more complete picture. And so I'm showing this because I want to summarize by observing that as trends in genomics, not just for the plant-animal space, but also for the human space and very generally, what we've been seeing, of course, that the workhorse over the past 15 years has been short read sequencing, and for good reason. It had the throughput, it had the cost associated with it. But what I would argue is that over the last 2 or 3 years, through the use of PacBio hi-fi sequencing on the Sequel IIe system, we've seen really how incomplete that picture was, how many gaps there were, how many errors have been made that now need to be corrected, and how many artifacts have occurred in all these different omics areas. And so with hi-fi-based sequencing, we now have seen what is possible to put together genomes more comprehensively, transcriptomes, epigenetics, and single-cell cell atlas. And now with the Revio system, you don't have to compromise anymore between data quality and scale and throughput and cost. And so we are very excited about bringing the system into your hands. And so very fitting and very timely, last week you may have seen the Nature Methods issue where we were very, of course, honored to see long-read sequencing being declared as the method of the year. Now the selection was made, we know that the selection was made and the content was generated before the Revio system was announced, and it was really fitting to see because the wish list by all of you in these articles is the data quality is great with hi-fi, just give us more reads and make them cheaper. And so we felt that this was an incredibly good timing with regard to fulfilling those wish lists. The other reason I show this slide is because I want to use it as an acknowledgement slide to all of you. No method is worth anything if the community is not adopting it and is not using it. And so we are so indebted and so grateful for all of you using the method, adopting it, pushing it forward, pushing us to make it better, and it's just been an incredible partnership, and so I wanted to thank you for that. As a company, we strongly believe that it is important that the different sequencing companies release data openly and publicly so that you can take a look for yourself. And so we had previously deposited human data sets for this conference. We have deposited freely and openly for use plant and animal data sets, as well as the MAS-seq data sets that I mentioned. Now, lastly, of course, there are some applications that do not require long reads. And so different capabilities are required from a sequencer when, and I'm just showing 2 examples here, when you look at literally millions of samples with genotyping by sequencing, finding needles in a haystack from the viewpoint of having lots and lots of individuals that you want to screen. And then in the gene editing specificity analysis, so our next speaker, Gina, and her colleagues had a paper looking at off-target activities from gene editing, and you really want to sequence deep and see something that may happen at a very low frequency, so again, a needle in the haystack. And so for that reason, we announced a more accurate short-read sequencer, which we call ONSO, that will be a mid-throughput sequencer, about 400, 500 million reads, the typical 2x150 paired end, or 1x200 or 2x100 single-end reads. And the third bullet is the main differentiation, which is that 90% of the bases are going to be Q40 or greater. So an order of magnitude greater than what you're used to from Illumina sequencing. And so for these needle in the haystack applications, what that means is you can sequence deeper and see the low-level variants with confidence. Or you can also not require UMIs. And so you don't need as much sequencing coverage for certain applications, which then makes it cheaper. The beta has commenced at 3 different sites, which are listed here. We're now accepting orders starting this week and we are expecting it to ship towards the end of the first half of this year. So with that, I will close and say that we are so excited to bring the new capabilities of the Revio system on the long read side and the ANSA system on the short read side into your hands. What you see here, enabling the promise of genomics to better human health, is our mission statement of our company. And I can tell you that we interpret human health very holistically. We believe that human health is not possible without a healthy and biodiverse environment. We believe that humans cannot be healthy without a stable and healthy and nutritious food supply. And so we really believe that that is all-encompassing. And so what these instruments will do is scaling this paradigm-shifting performance that I told you about by providing extraordinary accuracy, but now at a scale and a throughput and a cost a point that can make lots and lots of more projects possible.
All right, thank you, Jonas, and thank you to the entire PacBio team who invited me to speak here today. I am Gina Zastro-Hayes, and I am from Corteva AgriScience. So many of you, because this is an agriculture meeting, likely know who Corteva is, but just as a reminder, Corteva is a pure-play agriculture company focused on both seeds and crop protection chemical solutions for our farmers. And Corteva stands for Core, Heart, and Teva, Nature. But just because we're short of 5 years old doesn't mean that we are new to the marketplace. We are rooted in a long history of agricultural excellence from DuPont, Dow, and Pioneer. Those three companies came together in the mid-2010s to merge a single Dow-DuPont company and then spun three individual companies, Dow, DuPont, and Corteva, which is all of the agricultural components of the three originators. Our purpose as Corteva is to enrich the lives of those who produce and those who consume, ensuring progress for generations to come. We are committed to not only making our farmers successful today, but making sure the land and our farmers and consumers are successful for all future generations. And to deliver on that promise and our purpose statement, we stand by our brand values, which include enriching lives, standing tall, being curious, being upstanding, and living safely. And most important to my message here today, build together. So we not only do a lot of internal research work hard with all of our colleagues there on our individual campuses, but we reach out to the academic community. If you haven't had a chance to stop by our Corteva booth over in the showroom, we have an open innovation platform that allows you to interact with our company in the form of grants and special call projects. And we're interested in hearing in what you're interested in and how we could work better together with you. We also have a vested interest in partnering with our technology providers and vendors to make sure that they understand what we need to deliver on our missions for our customers and those end consumers. And that is why I'm here today. We partnered with Pacific Biosciences when we heard there would be coming a new higher throughput instrument to deliver on workflows that could feed that instrument in the end. So just a few more stats about Corteva before we get into the science behind what we're doing with PacBio. We have over 20,000 colleagues in our company. We do have over $14 billion in net sales and work in over 140 countries on many crops and crop protection chemicals that apply to all areas of agriculture. And our agricultural pipeline is built on innovation. So both our seeds and crop protection chemical business relies on a robust R&D pipeline to deliver to those end consumers. And when you really get down to the core of our company, we're really delivering DNA solutions to our customers. In seeds, that's pretty obvious. The genetics, the germplasm, transgenes, gene editing, those things all can be clear. But our crop protection chemical business is also driven by genetic gain in fungal and microbial sources for many of our crop protection chemicals and in our natural products discovery pipelines. I am lucky and have the privilege to be the leader of the Genomics Technologies group. And we have what we call our Corteva Genomics Toolbox, which includes all of the types of technologies you see at this meeting. Your short read sequencers, optical mapping technologies, nanopore sequencers, and also, of course, long read sequencers in the form of Pacific Biosciences. We have built a core reference genome production pipeline and have collaborated with many of you in the audience on that pipeline. And our pipeline is built on the fundamentals of hybrid scaffolding. So the vast majority of projects that come into our lab for reference genome sequencing start with DLS mapping or bionano optical mapping. Then we move on to hi-fi sequencing, which Jonas already touched on earlier. And then finally, hybrid scaffolding those into T2T end product pseudomolecules and genomes. Because of the advances in the technologies over the last few years, it's not a surprise to this particular group of people that we've had huge advancements in the contiguity of our overall genomes over time. So in 2016, if we use maize as an example, which has a variable genome size, but roughly 2.3 to 2.4 gigabases in total size, not too far off from the human genome, we were averaging 50 hybrid scaffolds per chromosome. By 2018, we were at 3 scaffolds per chromosome. But with the addition of hi-fi sequencing to our pipeline, we were able to move to less than 2 scaffolds per chromosome by 2021. So if you think about that from a particular, from gains, 5-year gains, 25x in contiguity, 40x reduction in cost, 15x reduction in time. As Jonas pointed out earlier, when asked what we wanted, the answer was more of what we're already getting. We could see that this technology could drive us in the right direction for the goals that we want to achieve. Why? Is that these genomes form the foundational data that we need for our predictive breeding programs. So that foundational data, if you think about a closed breeding platform in corn, we have a limited number of circulating haplotypes in our breeding germplasm. We want to sequence all of those potential haplotypes, and we can use that as foundational knowledge to build haplotypic frameworks and ultimately feed our prediction pipelines with lower density genotyping. That's not just for corn. It's for all of the crops that we work on, including projects we do for external entities. So the last time we were at PAG, now, rewind, everybody remember 2020, then there was a break. We were talking about our work with PepsiCo and the release of the oat genome, which has been released to the public shortly after PAG in 2020. So the technologies that we're applying don't just apply to maize, they don't just apply to soy, they apply to large and complex polyploid genomes as well. So what is the future vision? What are we trying to deliver at Corteva? Well, this is our 2030 goals, which is part of our sustainability package, and that is to increase crop yields for our farmers 20% while also reducing greenhouse gas emissions by 20% within cropping systems when compared to 2020. So this is our 2030 goals. How are we going to deliver on those goals? That's by helping farmers create resilience using multiple applications. So a big component of that is, of course, our R&D pipeline, which includes transgenes, gene editing, all of the technology-based solutions. And you'll see here in our slides, PacBio was actually focused on in those publicly facing sustainability slides from last year. We want to improve genetic gain. Obviously, I mentioned increasing yields for farmers. And then we also want to apply precision agriculture, specifically in our crop protection chemical solutions, and integrated solutions with digital solutions for our farmers to help manage their land. How does Revio fit into this? Well, as I mentioned earlier, we really need almost an infinity amount of sequence data to deliver on our goals for both breeding and the crop protection pipeline. And when we heard they were going to be launching a higher throughput instrument, our first thoughts were, that's great, but how will we feed it? Anyone in here that works on plants knows that plants have inherent qualities that make them both wonderful and also a little bit, tough to deal with. That cell wall sometimes when you're trying to deliver data in a high throughput manner doesn't really help with extraction. So we started coming up with a list of things that were going to be barriers to us feeding a high throughput system. Number one, unique requirements for plants and high molecular weight DNA extractions. Also, you can include unique requirements for microbes, for highly diverse microbe sets. We've also been working on high throughput microbe workflows. Library prep DNA requirements. We all know that the Pacific Biosciences platforms have higher amounts of DNA required for genome assembly than some of our short read platforms. and then low throughput processing steps that existed in the current SmartBell kits, and that includes shearing, size selection, and quality control. This is where our partnership with PacBio came in. We looked at the overall workflow and said, where are the places that are going to be roadblocks that I just mentioned? And how can we throw out the assumptions we've always had about these individual steps and find innovative or solutions that already existed in our labs to apply them to the PacBio ecosystem and create a high throughput process? So just starting with the first up front, tissue disruption and DNA extraction. In the past, many of us who've worked on plants have been using liquid nitrogen and old grinding techniques, and I'm sure all of us have had wonderful afternoons in the lab, grind away to produce enough high molecular weight DNA for a variety of applications. We threw away our thoughts on how we're going to treat that, and Charlotte Harris and Victor Laka and their teams in our lab, along with PacBio, found multiple solutions, primarily using genogrinder-like applications with beads and specific cooling steps that would allow us to, in parallel, isolate high molecular weight DNA for the hi-fi system, 96 samples at a time. That's at the level we can start talking about feeding the Revio system, 96 samples at a time. We also created, specifically Victor Laka's team, along with the Circulomics team, worked to use specialized solutions from the Circulomics portfolio, which is now part of PacBio, their buffer systems and their knowledge in extraction, using magnetic bead technologies to create parallel solutions for 96 well-based extraction for plant tissues, again, enough to feed the Revio. So here's some examples that were generated during the pilot projects of a variety of plant tissues, the input tissue amount, DNA yields, and the sizes, all sufficient for use on hi-fi, all producing enough DNA for us to proceed with that library prep. The next is DNA sharing. So we have the hi-fi DNA. Now we needed to get it down to the size that will work for the hi-fi library prep. Again, many of us have been using systems that can produce between 1, 4, or 8 sheared samples at a time. That is not going to be able to feed the Revio, which can produce 15x the amount of data we've been used to on our Sequel IIe systems. Now have 2 different validated methodologies. One, again, based on genogrinder-like bead-based shearing or agitation, and one based on pipette-based shearing. So these are using common pieces of lab equipment, not some fancy solution that you've never heard before. Again, throwing away the assumptions about the types of techniques we had to be developing to deliver on the Revio system, and they are also fast, less than 10 minutes for 96 samples, or 15 minutes for 96 samples, depending on the instrument, and easily able to implement. And you can see by the size graphic on the below there, on our traditional Megaruptor systems, which we've been using for years with great success, same sizing essentially with both the plate-based shearing system agitation and the pipette-based shearing system. Next was library prep, and PacBio actually came with a solution, and that was the SmartBell 3.0 library system. Many of you have likely tried this already. It is very amenable to automation solutions. So PacBio already has partnered with Hamilton to create solutions for 96 libraries in parallel. This is also amenable to normal lab processes with plates and multi-channel pipettes, multiplex, all the things that we're used to, I would say, on our short read platforms delivered for the hi-fi platforms. Even more impressive is the bead-based size selection, which allows us to remove small fragments and maximize the number of hi-fi reads we want in that 12 to 18 kb range. And then finally, sequencing. So all of this is great, but it's only great if it really delivers the same hi-fi, great quality data that we're used to from the Sequel IIe systems. And when I was talking to my leadership about these systems, the one risk was always, well, it's hi-fi chemistry, but we haven't seen the instrument, it's unknown. We've all purchased instruments, we all know there's always that risk. They say it's gonna work, but we don't know. Jonas already showed this data. We feel very, very confident that this is going to deliver on all the promises that were already, said, 15x the data with all of the quality we're used to from HiFi. To test this, we have sequenced 2 individual maze lines here and taken them all the way to assembly. We have also sequenced 96 barcoded maze lines as a pool and showed that pooling and barcoding strategy does work successfully, so we can start thinking about this as a resequencing application. So no longer missing all of the large structural variants in our resequencing data, no longer wondering about repeats in our resequencing data. This is HiFi data for all of your resequencing needs, which is really amazing. And this is, Jonas already showed in column 1, our Maze 1 reference data. This is actually a B73 sample. We are working to allow this to be released as well to the public, so you'll be able to take a look at this data. What's the most exciting is that in the hybrid scaffolding step, when we took this new Revio HiFi data, assembled it with our normal assemblers, and then did a hybrid scaffolding with BioNano, we saw either improved total number of scaffolds or roughly the same. So we feel very, very confident that the HiFi data being produced by Revio is at least equivalent, if not better. In maze number 1, in that particular example, we had a significant reduction from 22 scaffolds in the Sequel IIe version to 15 in the Revio version. When we took a look at the individual read quality, we did see the improved read quality that we were expecting with deep consensus, and we anticipate that is why we're seeing the improved overall assembly results in this particular sample. So just to summarize, Revio HiFi sequencing from our early pilot programs is equivalent or better to what we are seeing on the Sequel IIe. The initial Revio data is meeting or exceeding the requirements for our plant genome sequencing and assembly. The new high throughput workflows that we co-developed with PacBio are complementing the capabilities for Revio and really allowing us to move to new application spaces that we didn't think would be possible for long read sequencing for many years to come. And as a reminder, all of this comes along with at least the 5 MC data in the background. So you can start thinking about interesting questions like, what if I made multiple tissue libraries and created a reference genome that didn't represent just a single sample, but multiple samples, and have that methylation data along for the ride. As Jonas mentioned, we are also an ONSO beta test site, and we have completed some pre-beta testing on site at PacBio with some of our samples. 10 maze samples sequenced single read 200. We've completed that data analysis at Corteva and compared it to our reference data sets. And then we also have a new ONSO instrument installed in our facility, and training was completed last week. Beta tests using both conversion and new library prep methods have begun, and we're excited to really put that instrument through its paces. So what are our initial thoughts on the system? We are seeing increased accuracy, as Jonas indicated, but I think the most exciting has been improved handling for a short read system of poly-C, poly-T, and poly-AC homopolymer and dipolymer stretches. So example here I have is a poly-C and a poly-T. On the top panel you have ONSO. We are seeing some reduced coverage in these regions, but we're not seeing the data fall apart. We completely lose it with traditional short read platforms. We're not seeing that. We're also seeing very accurate overall count numbers of these homopolymer stretches. If any of you have ever done sequencing for regulatory, you'll know that they will ask over and over and over again, is that 29 Cs or 30 Cs? And you can ask that question 100 times and come up with both answers, 50 times each. So this shows really new potential for us to answer some of those questions in these discrete regions that are important specifically for regulatory questions. So I guess I'll end this by saying we believe that these new platforms offer really great potential in agricultural genomics, and they allow us to keep growing. And with that, I'd like to thank both the Corteva team and the PacBio team who worked on this project.
So I'm going to talk to you today about something which is a little different, and it has to do with using both long-read and short-read data and genomics data for conservation work, especially here in California. And before I get started, I just want to say I'm not going to be doing a lot of sort of promoting of PacBio in this talk, but Jonas in particular and the PacBio team in general have really helped us tremendously with this project, the California Conservation Genomics Project, in really meaningful ways. I mean, we've gotten some breaks on chemistry, and that's great, but what we've really gotten is their brains and them helping us as we've done our genome assembly meetings, sometimes every week, sometimes every month. They've been there, they've been participatory, and really helping a lot in really meaningful ways, and I'd just like to thank you for that. Okay. So let's get started. So I have a few different goals, and really, I think as much as anything, what I want to do today is tell you about a specific project that we're doing, this California Conservation Genomics Project, and also just impress upon you the fact that there really is, I think, a very productive meeting of the minds between genomics and conservation. And we need both if we're going to save even the remnant scraps of biodiversity that we have left in California, in the US, and in the world. So I'll talk briefly about the flavors of genomics for conservation, that is neutral versus non-neutral variation, why at least in our view, our collective view, business as usual, the way we've been doing this kind of work, which has been going on for decades, really isn't enough, how this California Conservation Genomics Project is changing the game, and then spend most of our time on progress. So one slide on sort of why this is important, and hopefully many people in this room already know this, but if we think about variation, and this is sort of thinking about population genetics and population genomics that's out there in the real world, we can think about neutral genetic variation, and we can think about genes under selection. And neutral variation informs us about, in fact, interestingly enough, most of the things that the conservation community cares about traditionally. It tells us about population structure, historical gene flow and current gene flow. It talks about hybridization, which can be a big issue in conservation. It gives demographic inferences, things like effective population size, which it turns out now is much easier to measure in many cases with natural populations than it is to just go out and count things, because counting stuff is hard. And it tells us, importantly, about levels of standing genetic variation. As populations become isolated, they become inbred, they lose their genetic variation, they lose their genetic resilience, we get inbreeding depression, start going down sort of an extinction vortex, as it's known. And we can measure that and quantify that and hopefully deal with that using neutral variation. There's also a whole other world, which has always been sort of invisible to most of the natural population community, and that's studying genes under selection that tell us about adaptation. But if we want to really deal with climate change, which is the big bugaboo that everybody is worried about, we need to understand those genes that matter. We need to understand genes that are important in local adaptation and selection, and we need to deal with them. So what is the community currently doing? I would argue there's sort of two different approaches to conservation genetics and genomics that are ongoing in the community right now. The first are some of the large multi-species projects that you've hopefully heard of. There have been talks about them at these meetings, things like the Vertebrate Genome Project and the Earth Biogenome Project that Harris Lewin headlines. And basically, they're all about producing lots and lots of reference genomes. High quality reference genomes, really important reference genomes, often using PacBio long-read sequencing. Usually a single example per species, okay? The other side of it is sort of single species population level projects. So that's sort of the purview of standard population genetics. We go out and we get a bunch of turtles or salamanders, which I happen to work on, or oak trees, or whatever else it is. We sequence them from across their range, and we learn something about that specific species that might be important for conservation. Often, the community uses reduced representation approaches. RADseq is the common one that everyone's using these days, ourselves included. And it allows us, because of the cost structure, to do population level sampling. So just to give you a sense of this, I asked a colleague, very close colleague of mine, a woman named Kat Darst, who's the head of listing and recovery for the US Fish and Wildlife Service, about a year ago. And I asked her the basic question: if we had a high quality reference genome from a single individual for every plant and animal species in California, would that change what the US Fish and Wildlife Service does when they get up out of bed in the morning? And remember, they're the group that lists species under the US Endangered Species Act. And Kat, in her kind of classic way, said, "Hi, Brad, it would be a little useful, maybe, to identify candidate genes for different features. Maybe, but our bread and butter is within species stuff, intraspecific variation and species delimitation." And so hearing what we just heard in the last two talks about the possibility of doing long read sequencing at scale for population level work and at cost, to me, was huge. It makes me want to sort of take the last 4 years of work that I've done and throw it all in the toilet. But we're not going to do that. But anyhow, that's kind of an important thing to remember. So what it means is projects like the VGP and the EBP, they're assembling these absolutely critical resources using PacBio information and PacBio technology, among others, that are needed for conservation, but it's not the full data that managers need. We need that population level stuff. So let me just give you one quick example. I'll just run through this real fast. This is an example from a couple of years ago from our lab. It's a project we did that just shows you what can happen in the sort of space of conservation genomics. This is a foothill yellow-legged frog, a species that's basically endemic to California. We did a population level study of it using RAD-seq data, about 40,000 loci, from 50 localities across about 100 frogs. And what came out of that study is, you can sort of summarize it as this. I don't know if you can see the colored dots on the map, probably not. But basically, across the whole range of the species, and the range is not showing up here, which is too bad, what resolved from this was a set of 5 very, very well-defined clades of these frogs, ranging across, covered across the range. And the state of California was trying to figure out what to do about potentially listing under the California Endangered Species Act this species of frog. And so what they did is they used that population level genomic data, RAD-seq data, to come up with a sort of structured listing where some populations were listed as threatened, some were listed as endangered, and some weren't listed at all because they seemed to be healthy. And so what it basically did is took this information and a very thorny, difficult problem in the regulatory world and turned it into this, where it said that the 3 clades in the southern part of the range were all listed as endangered, critically endangered, demanding a lot of work and attention. 1 clade in the northern Sierras was listed as threatened, not as at risk, but still at risk. And then everything pretty much from the Bay Area north was not listed, so not under regulatory control. A very kind of creative, interesting response based on these kinds of data. So that's just an example of how these kinds of data can actually work with agencies and should work with agencies to really give them guidance on what to do. So where do we stand? In my view, at least, we stand at a place where we're oftentimes developing fabulous reference genomes for many, many taxa. We have partial population level data, reduced representation data like RAD data, collected in sort of an idiosyncratic way for some T&E, threatened and endangered species. But every one of them is different. Every one of them is sort of idiosyncratic. And that makes it very, very hard to compare these studies and to sort of come up with any kind of a consistent population level analysis of a landscape, like, for example, the state of California, or communities, like, for example, the intertidal. We have no real way of compiling that data in a meaningful way because it's all collected in different ways. So, for example, this is a spadefoot. We already saw one, an example of one. We have some great RAD-seq data on this species. We also have some great Brad-seq data on this, the only freshwater turtle in California, but they're so different that you can't even compare them. Different depths, different sequencing strategies, different everything. I'd like to be able to include some flowering plants and compare those to that frog and to that turtle. I'd like to compare butterflies. I'd like to compare, in fact, all kinds of species. I'd like to be able to look at the whole swath of California biodiversity. I'd like to be able to look at hundreds of species: marine, terrestrial, freshwater, vascular plants, invertebrates, a whole mess, and ask, what can we say about the landscape genomics of California? And amazingly enough, in 2019, we convinced the state of California to put in the 2019 state budget a line item to do just that. And that was called the California Conservation Genomics Project. We're now about 3 years into that project, and I want to tell you a little bit about it. So the CCGP, as we call it, at a glance, 250 species covering all the ecoregions of the state, all the different taxonomic groups in the state, which are spread across 150 projects. And a project is either one species or 2 or 3 closely related species that we consider as one. For each project, we put together a reference genome, and that's where we use our PacBio data as well as OmniSeq data for scaffolding. We also, for each of those 150 projects, resequence basically 150 individuals that are spread across the state of California. Okay, they're all analyzed in a uniform way. They're all done at 10x coverage. Everything is whole genome resequencing. Everything is mapped back using the same protocols, the same filters, the same bioinformatic pipelines that we developed to give us a very consistent and comparable analysis that should allow us to sort of generate a map of genetic variation in California. Where are the pockets of high and low genetic diversity? So the goals of the CCGP, and we're now in year 4 of it, are really sort of a couple. At the individual level, we want to think about things like what I showed you for that foothill yellow-legged frog. We want to understand how we manage individual species that are of conservation concern or that are ecologically so important that other conservation concerned species rely on them. We want to understand where's the population genetic structure, where are pockets of high and low genetic diversity within species. Is the taxonomy that our conservation is based on real data. We can also then take all those species, because all the data are collected in the same way, compile them into a multi-species analysis and look uniquely for regions of high genomic diversity and high genomic resilience. All other things being equal, if certain mountain ranges, certain places like, I don't know, the Tehachapi's, for example, harbor high levels of genomic variation in species after species after species, those are the places that are going to be probably most resilient to climate change. And those are the places that the state and the feds should be protecting for biodiversity. And if they aren't, we can at least point them in that direction. And then both at the individual and multi-species level, again, we can look forward to analyzing genes that are important for local adaptation, and we can do that because we're using whole genome resequencing. Our team is really big. It's 79 principal investigators. It's UC focused, University of California focused, so it's all 10 UC campuses plus lots of other collaborators. We've got an executive committee, a few hundred co-PIs, and we also include the California Department of Food and Ag to look at pest species. The details of it, this project was funded at the level of $12 million by the state. All the data have to be consistent. As I said, that means that the reference genomes are completely done by us and our team. Merle Escalona is, I think, in the audience somewhere. There she is right there. She is the reference genome assembly team. Go Merle. And then our resequencing is done to sort of maximize landscape coverage across the state and apply that to areas of conservation relevance. So how are we doing? Let me just run you through in the next few minutes some of our progress. At the reference genome level, We have actually 153 projects that we need to develop reference genomes for. 5 of those already had good reference genomes, so we need 148. We have the tissues in hand for 145 of those. We have moved through our Hi-Fi and OmniSeq sequencing for most of those, or an awful lot of those. We have, you can sort of see the numbers here. We have scaffolding complete for about half of them. We have genome assemblies complete for well over a third. And we've published papers on, at this point now, probably a couple of dozen of them as genome release papers. So we're well on our way. And I think within the next few months, we hope, we'll be done with the bulk of the reference genome resequencing. In terms of phylogenetic coverage, this is something that I thought was kind of fun. We published actually an entire issue of the Journal of Heredity. The last issue of 2022 was dedicated to the CCGP. And Aaron Toffelmeyer, who's our associate director of the project, was the lead on this. What we did was put together a time tree, a time-calibrated phylogeny for every family of multicellular organism found in the state of California. And then we asked which of those families are included in the CCGP. And in terms of the phylogenetic diversity, or PD, which is really just sort of the sum of the branch lengths of that time tree that are included with our taxa, how are we doing? And we've got something over 15% of all of the phylogenetic diversity of multicellular life in the state covered in this tree, so in our project. So I feel pretty good about that. How do those reference genomes look? This is a couple of Murley's plots. Here's a western pond turtle. You can see some very good numbers. Contig N50 of 75 megabases, scaffold N50 of double that. Busco scores well north of 95%. Feeling very, very good about most of these genomes. They're not all that good. This is the first one I think we published, the Big Berry Manzanita. It's a plant. You may love plants. I hate plants because never will I ever work on a plant again in my life. Anyhow, but plants work. The numbers are still very good and something to be proud of. In terms of our whole genome resequencing progress, that's a little slower because there's a lot more to be done. The first thing we have to do in the whole genome resequencing, and this is something you might not normally think about, is figure out, we can pick 150 individuals, and we want to cover the ecological and geographical breadth of the state of California for each species or species project. How do you do that? And this is a project that Ian Wong, our lead on the landscape genomics, faculty member at UC Berkeley, came up with. On the right is a principal component analysis of the environmental space in the teal of California, basically precipitation and temperature. And on the left is a geographical map. And what we've done is for every proposed project with 150 samples, we've mapped them both in that environmental space and also in that geographical space. And you can see in the top one for the western fence lizard, a wide-ranging species, we have very good coverage of both the ecological or environmental space as well as the genetic space. The only place that we're sort of missing is that lower right-hand part of the environmental space or geographical space, and that's the deserts, and western fence lizards don't live there. They're replaced by a desert specialist. The western spadefoot is a vernal pool specialist amphibian. It has a much narrower range, but at least within that narrow range, we have it covered both environmentally and geographically. In terms of our overall sampling and our overall coverage of the state, we can break this up across the state and across the 19 ecoregions that are usually defined by the state and see how we're doing. These are the samples that we have in hand, and you can see each dot is a sample. We have the marine inshore community covered very, very well at the level of individual samples. We have plant coverage across most of the state with a hole in the Central Valley, but that's all agriculture, so it's hard to beat that. Vertebrates are even better covered across the state of California, and even terrestrial invertebrates, which are always a little bit difficult, we have pretty good coverage of. So we're feeling, again, very good about that. At the level of the ecoregions of the state, again, this is our coverage. These aren't all done yet in terms of numbers of individuals in black and number of species in red. And those are just sort of mapped onto the 19 ecoregions of the state. And what you can see is that both at the individual level and particularly at the species level, we've got great replication. So if we wanted to compare, for example, coastal San Diego County, right along the coast, to the immediately inland peninsular ranges of San Diego County, you know, we'd have something over 100 sets of species that we could compare and over 1,000 individuals that we could compare if we wanted to look at, say, patterns of biodiversity across that range. So again, feeling very good about that. At the whole genome resequencing level, again, our goal is roughly 20,000 samples across all those projects. 95% of those are now in hand and collected, so that's great and a huge challenge. About 40% of those, a little bit more, this slide's a little out of date, have been prepared. About 30% of them now have been pushed through and have data. And we have 26 out of those 150 with both data sets that are being pushed through the final pipeline. In terms of data analysis, again, what we wanted to do is make sure that all the data analysis for us, for the CCGP, is done in a very, very consistent way. And so the way we've done that is we get the data, we push it out to the PIs and say, do whatever you want with it, you know, follow your dream. But from the CCGP side, we've hired postdocs and identified, usually a young professor, to lead those postdocs. And we have an informatics team at UC Santa Cruz for the resequencing data, led by Russ Corbett-Dedig, and postdoc and a couple of data wranglers. And we also have a similar project and team led by Ian Wong at Berkeley with postdoc and grad student. The informatics team is very well established and really working things through well. They take in an assembled genome that Merle gives us, the whole genome resequencing data, and what they push out through a very kind of economical cloud computing framework that's really quite cheap is a clean VCF file that they can do in under 2 days for that whole set of data. On the landscape genomics team out of Berkeley, we have a pipeline that's been developed de novo from scratch that you can put in the data for individual species. It conducts several standard analyses in R. It outputs beautiful quality publication figures and maps, and it can take in any SNP data set. And I'm a herpetologist, as you might have figured, because all my examples are reptiles and amphibians. And coincidentally, it's called Alligator, is the name of the project, of the software, which is really great. Real quickly, I've got a couple of minutes left. This is just to show you, and this is just sort of the most basic output that actually is part of our QC project or protocol that comes out of the informatics team. But it's just to say, if, you know, maybe the most basic thing you can get out of this kind of data are what are the units of differentiation, you know, based on neutral loci across the landscape. And again, this is a ruby spot damselfly, a very common species associated with aquatic habitats. And basically, it's got 3 different sort of clades, 3 different lineages, with a contact zone between them just north of the Bay Area and separating the Central Valley from the coast ranges with a distinct additional set out in the drylands further east. Well, if you wanted to know, is that repeatable? Do other insects show that? Do other flying vertebrates show that? Who shows that and exactly what way and how? Again, we can even just visually, without doing the complex analysis, start stacking those up together. And what you can see, I just sort of threw these red lines on these maps at this very, very simplistic way, is that there's similarities, but they're not identical. There's oftentimes a coast range clade that's different from a Sierra and plus Northern California clade. Northern California almost always goes with the Sierras and not with the coast ranges. But the details of those breaks, if you're wanting to manage the song sparrows on the right, you know, compared to say the ruby spot damselfly, are subtly different. And that's where this comparative data set allows us to really shine. We've completed 12 projects. We're going to be completing a whole lot more of them. My sort of modest goals are to have the reference genomes done by the end of this winter and the whole genome resequencing work done and analyzed by the end of 2023. Lots of acknowledgements, including our pals at PacBio. But the big acknowledgements, and this certainly includes Murli, that I just want to say, and then I'll step down, is this team of people. And if you look at them, these are the folks that are doing the analysis, that are doing a lot of the bench work. We've set up a mini core facility at UCLA just expressly to do this work. They're all young, they're all enthusiastic, they're all totally driven by conservation, and they're a real joy to work with. So I'll leave it at that.
All right, I first would really like to thank the organizers of this session for this opportunity to present some of the work that I've been doing. My name is Sarah Carey, and I'm a postdoc with Dr. Alex Harkis at the Hudson Alpha Institute for Biotechnology. And I'm going to tell you about some of the exciting work we're doing on assembling plant. We love plants. Plant sex chromosomes. And so today what I'm going to tell you about is my favorite trait in plants. And because plants and animals and other systems use different terms, I want to start off with kind of walking you through some terminology. My favorite trait in plants is something that's called dioecy. And so in plant world, that is the separate sexes. It's the same thing as gonochorism in animals. But for plants, what that means is that carpels and stamens develop on separate individuals. And so this is something that's actually pretty rare in flowering plants. Most flowers are what's called perfect, where the flowers have these whorls of sepals, they have petals, they have the carpels, and they have the stamens. With dioecious plants, instead, females and males will both have those sepals and those petals, but only females will have functional carpels, and only males will have functional stamens, so separate sexes. And whether you're aware of it or not, you've probably interacted with some dioecious plants, especially if you've hit the monkey bar this weekend. If you drink things like yerba mate or sarsaparilla or beer, the hops that's in there, those are all dioecious species. They have separate sexes. Your CRISPR probably has some dioecious species in it, so if you eat things like asparagus or persimmon, I heard persimmon earlier, that's dioecious, or mulberries. Your spice cabinet's probably also full of dioecious plants, so things like allspice, your black pepper, and Sichuan pepper all are dioecious. And so one thing that we're really excited about trying to get after is understanding how many times dioecy has evolved in flowering plants. So it's super rare, it's in 5% of plants, but how many times has it evolved? Is it hundreds? Is it thousands? And when we think back to, I mentioned that there's pepper, it's literally peppered across the phylogeny. Get it, pepper? Ah. Thank you. I'll add in a couple more of the species that we've been talking about. And so what this is showing is the 64 angiosperm orders, and a few of those choice species that we've been talking about. In gray are orders that have no known dioecious species. In yellow are there more dioecious species. Blues are fewer. And even from this scale, you can see that there are some clades that don't have it, so there's different evolutions. But as we start moving further and further in and looking closer and closer within a family, within a genus, we start finding more and more and more. So looking more on the side of thousands of evolutions. And so the other thing that we're really excited about going after is figuring out what genes are associated with these transitions from having a perfect flower to having separate sexes. Because that gives us a catalog of novel carpel and stamen sterility genes that we can use in breeding programs. And so, unlike animals, where there's kind of some key players that typically show up, like SRY or DMRT1, so far in plants, everything that we have looked at has had a different set of sex-determining genes. This has only been examined in very, very few species with functional validation, but of the ones that have been, on the left, those are the carpel carpel sterility genes, and on the right are the stamen ones. And what I want you to take away from this is they are all different genes, and they often hit very different parts of the developmental pathway. So how many other genes are there in these hundreds to thousands of evolutions? And so because dicey is the evolution of separate sexes, it also tends to have an evolution of sex chromosomes. So that's a really, really nice thing that takes our catalog of genes that we're trying to figure out that are associated with carpal and stamen sterility down to one sex chromosome in that non-recombinant region. So one bit of a genome that we need to examine genes within. And so sex chromosomes have particular characteristics. The Y chromosome has this non-recombining region, and within that should be where our sex-determining genes are located. The problem is, sex chromosomes are notoriously complicated to assemble. If you've been following the human T to T assemblies, you may have noticed that just last month, the bioRxiv preprint for the Y chromosome was released. That Y chromosome, even though it's a small chromosome, was the hardest and the last one to be able to put together. And that's because they're just very complicated parts of the genomes. If you look at the autosomes and the X chromosomes here, you can see there's lots of gene content. There may be some heterochromatic regions, but for the most part, lots of genes. But that male-specific region of the Y chromosome, that non-recombining region, very gene poor, tons of heterochromatin, super repetitive. That's historically made it very tough to assemble contiguous genome references. And so what we are doing in our lab is we are assembling genome references of these complicated sex chromosomes, and what we want to be able to do is have completely phased haplotype assemblies. We ideally want this for autosomes and sex chromosomes, but I'm focusing just on the sex chromosomes today. And so what we want is to have contiguous assemblies for both our X chromosome and our Y chromosome, so we can get those sex-determining genes. What tends to happen, though, when you do a de novo assembly is you don't know which came out in haplotype 1 or 2. Do you have the X chromosome or do you have the Y chromosome? Moreover, what can happen is you end up actually having a chimeric mix between the X and the Y on both haplotypes. So it's the same thing as thinking about maternal and paternal haplotype switching in your assemblies. That can happen to the sex chromosome, and that makes it very tough to figure out which piece you're actually looking at between those two haplotypes. And so this is something that we've been thinking about and working on figuring out how to tackle. And so we have this pipeline that we have called plant sex chromosome cytogenetics by sequencing. And if you aren't familiar, this is a picture of Dr. Nettie Stevens, who is the brilliant scientist who first discovered sex chromosomes in mealworms and published her work in 1905. And so we're taking inspiration from her brilliant work of finding sex chromosomes, but we're doing it using sequencing approaches. And so broadly speaking, the principal idea about how we're finding these sex chromosomes is by using sex-specific k-mers. And so if we think about an XY system, all those autosomes and the X chromosomes, they are shared across both males and females. But that Y-specific region, that non-recombining region, is only ever found in males. So if we find k-mers that are only found in males and never found in females, those belong to our Y chromosome. We can also apply this to the ZW sex chromosome system that is in things like birds and moths and some plants like Amberella. And if we find all those female-specific k-mers that are never found in males, those belong to our Y chromosome, so that non-recombining region. And so I'm going to talk about a few examples where we've used our pipeline to identify our sex chromosomes. And so the first one I'm going to talk about is Amberella trichopoda, which I lovingly refer to as the platypus of flowering plants. And there are a few reasons why. One is that it is a monotypic species, just like platypus. It is the sister lineage to the rest of flowering plants, so it holds an incredibly important phylogenetic position for comparative study. So it means we really want to have good assemblies for those kinds of comparisons. And it has relatively weird sex chromosomes. So Amberella has those ZW chromosomes I mentioned. Platypuses have something else going on. We won't go down that rabbit hole today, but we're just going to focus on AMBO. And so using PacBio Hi-Fi and Hi-C data, this is an assembly that was done by Jerry Jenkins out of Jeremy Schmutz's group. So this is part of the Open Green Genomes project. And Jerry and team were able to completely phase out both the haplotypes for Amberella, beautiful contact maps, all in 13 chromosomes like we were expecting, and contig stats look great. We have very few contigs, the N50s are really high, and both haplotypes have very similar length, so beautiful phased assemblies. So when I use our cytogenetics by sequencing pipeline, we can find that we also have a beautifully phased sex chromosome pair. So what we're showing here is haplotypes 1 and 2 by chromosome, and what are mapped are what I'm calling W-mers, so those female-specific K-mers, and you can see there is a single dense peak of them on chromosome 9. And if we zoom in on that region, you can see much more clearly that that is a contiguous piece of W chromosome, only in haplotype 1, and nothing found in that homologous region of haplotype 2. And so Amberella has a 3 megabase non-recombining region on the W, and what's really cool is that this assembled in a single contig. So we're going to talk a little bit about why that's really cool in another example, but the fact that we got through a sex chromosome in a single contig is very special. And so getting back to those original questions I was talking about, next thing we're doing is working on trying to identify the sex-determining genes in Amberella. There's about 100 genes on both the Y and, the Z and the W in the species, so might take a little bit of work, but that's what we're after next. And so this is a relatively small non-recombining region, relatively small sex chromosome. What happens when we look in a really old, really big, really complicated pair of sex chromosomes? Something like hops. So the next bit's going to be about some work I've been doing as part of my USDA postdoc on assembling hop genomes. And so hops are in the species Humulus lupulus. There's 5 varieties of Humulus, and it's largely the European variety, variety lupulus, that is used in beer. And it's only the female flowers. Those are called the hop cones that are used in beer production. Hops have an XY sex chromosome system, but what's really complicated building these assemblies and uncovering sex-determining genes is hops also have really big genomes. They're about 3 gigs. They're really heterozygous at about 4 to 5% het. And they're super repeat rich. I have it about 83% repetitive for the varieties that I've looked at. So complicated genomes. But despite the complications, I have 3 phased haplotypes to show you. So we have used PacBio Hi-Fi and OmniC data, and this is 3 of the varieties of hops. And something that's really interesting that happens when you have super complicated, super big sex chromosomes is that they both end up in haplotype 1. So hops have 10 chromosomes, and when you scaffold up haplotype 1 out of these assemblies, I have marked underneath them both the X and the Y chromosome end up in that haplotype 1. And we'll see why that happens in a couple slides, but that's just what happens. Haplotype 2 has 9 chromosomes. It's a little bit complicated. It's something that I hope we can figure out how to resolve in the assembly, but in the meantime, what I figured out is the best thing to do is just pull one of them, put it in haplotype 2, and scaffold that back up. So once we do that, we have 10 chromosomes in hap 1, 10 in 2. And here's some of our after scaffolding stats. So we're right at that 2.6 gigs for hap 1, 2.4 for hap 2. Most of the sequences in those 10 chromosomes, so 95, almost 99% in hap 2, and got some pretty good BOOSCO scores in these assemblies. And I'll admit, if you look closely at the contact maps, I have narrowly focused on the sex chromosome, so if you see anything else on those other pesky 9 chromosomes, it's a work in progress, but we'll fix those before release. I don't want to keep showing you tons and tons of contact maps, though, but I can tell you that the other 3 varieties, we did a similar thing, pulled one of those sex chromosomes out of hap 1, re-scaffolded it up, and everything looks really nice so far. And so once again, we used our pipeline to identify the sex chromosomes, and so haplotype 1 in the beer hop has the Y chromosome. You can see that giant peak of what I'm calling Y-mers, so those male-specific K-mers. And next to nothing in haplotype 2. And this slide really highlights why it is that both the X and the Y are ending up in haplotype 1. So I said the hop genome is pretty big, right? It's 2.63 gigs, 10 chromosomes. The sex chromosomes themselves are 167 megs to 250, depending on the variety. There's variation, that's very cool. But 80% of those chromosomes, 80%, is non-recombining male-specific region. So we're talking 135 to almost 220 megs, depending on which variety. So huge parts of these chromosomes look completely different from one another. And so that's why they're ending up both within one haplotype. But what I think is just incredibly cool and very powerful with these new technologies that we have is 220 megabase non-recombining region that's even more repetitive than the genome average. This SDR region is 95% repeats, assembled in 2 contigs. 2. That's like 2 Arabidopsis genomes worth of like non-recombining region that was pulled together in 2 contigs. I think that's just so cool that we're able to do this now. And what makes this even more special is, as I was kind of putting these talks together, is I realized that the hop sex chromosomes were discovered by Vinge exactly a century ago. He published his paper describing these exactly a century ago, and now, 100 years later, we finally have genome assemblies that we can start really, really diving in and looking at these regions more closely. Moreover, the hop sex chromosomes were some of the first pairs to be described as being heterochromatic. So back in 1957, so a few short years after being discovered, Jacobson showed that the Japonicus, which is the sister species to beer hop, is super heterochromatic, and using the 5MC calls from PacBio, we're able to kind of see that all the genome has some pretty nice levels of methylation, but I have an arrow pointing right at that sex chromosome, and you can just see its probability of being methylated is near 100 across the entire thing. And then kind of zoomed in on a particular region where you can see those repetitive pieces that have been annotated are super methylated. And there's a piece in the middle that once we get some gene annotations, maybe there's some gene in that blue region. We'll see. This is very, very early days, but very excited about the ability to be able to use these kinds of data from PacBio. So as I've kind of been saying, sex chromosomes are hard to assemble. There's still some work that we have to do, additional rescaffolding and other things. But just like humans, hops have these really heteromorphic, heterochromatic, repeat-rich sex chromosomes. So bringing it back, the plants and animals have very similar complicated parts of genomes. And just a very cool thing that I've been able to look at since getting these assemblies is back to that other question that I had of how many times has dioecious evolved in plants? Now that we have these assemblies, we can do things like run synteny analyses using something like GeneSpace to ask, is our sex chromosome pair the same as other closely related species? So there's an existing hemp genome that I've used here and a fig genome, and then the mulberry genome is a red mulberry that we've also assembled. And what we can see is I've highlighted the sex chromosomes in red, and hemp and hops are in fact the same sex chromosome pair. So within Cannabaceae, it's the same XY system. But figs and mulberries both have completely different chromosomes. Moraceae is sister to Cannabaceae, so they are fairly closely related, but it's really kind of interesting that they have an XY pair, both of them, but they are different chromosomes, which we're not sure yet if they are truly independent evolutions or if there's some translocations. These genes are jumping around across these different lineages, and it'll take some more finer work, but we'll get after that. But this really highlights that point of like, we've got to look at multiple different evolutions because there's the potential to find new different carpal and stamen sterility genes in these species. Okay, conclusions. So sex chromosome genome assembly, we can now phase sex chromosomes. It's really, really, really cool that we can do this now. K-mers are really ideal for finding those SDRs, and if you want to chat more about why I think K-mers over any other standard analysis, I'm happy to do so. Sex chromosomes may still end up in multiple contigs, and if that happens, they may split between the haplotypes. They can end up being chimeric or end up being shoved in one haplotype. So if you're running something that's dioecious, these are things that you really need to look out for when you're assembling sex chromosome pairs. We showed that Amberella has a fairly small SDR. It's about 3 megs with 100 genes. Hops have way bigger sex chromosomes, though. They're 167 to 240 megs, and they're the same XY pair as cannabis, but different than Moraceae. And so Next, we're working on identifying those sex-determining genes also. And we want to keep doing this. We want to sequence more dioecious things. Ideally, I want to sequence all of the dioecious things, but that's like tens of thousands of species, so we'll start with 100. But my postdoc advisor, Alex, just got news that he has been awarded an NSF career grant to sequence a bunch of dioecious species. And when we put this in, we actually were planning on doing 25 to 30, but then coincidentally and fortunately for us, Revio was announced, so now we're shooting for 100. So we can literally triple that, and we're very excited about that. So please feel free to reach out if you work on something dioecious and would be interested in collaborating with us on assembling some sex chromosomes. With that, I'd like to thank all of my amazing collaborators, my postdoc advisor, Alex, Haley Hale, Hannah Mueller, Laramie Kusbeck, and Zach Maharg. They are the ones who do all the wet lab work. I'm computational. And without them and their amazing skills in the lab, my assemblies would be much tougher to do. They're awesome, thank you so much. Jeremy Schmutz and Jane Grimwood and folks in their groups, Jerry Jenkins, John Lovell, and Les Gorton and Aaron Liston have been extremely kind collaborators with helping collect tissue for some things like the red mulberries. Josh Havel, Gary Muehlbauer, Catherine Easterling, and Nicolab, sorry, didn't put you on here, but all the folks at Hofsteiner and Josh Havel for helping me with the hop work. And then Adam Healy and Stewart for your work on sphagnum. And yeah, thanks to the postdoctoral fellowship from the USDA and Alex's NSF career. And yeah, I think that's my time. Thank you.
Great. Okay, so I am the last talk in the workshop today. I'm happy to be here. My name is Dan Portick. I'm a bioinformatics scientist. I work at PacBio on the computational biology team. And so we're going to shift perspectives now from the macro scale all the way down to the micro scale. And my talk today is going to be focused on microbiomes and metagenomics specifically. But before I jump into the main content, I need a little bit of help. So I need to take a quick poll. Quick show of hands, how many people in the audience have worked on a microbiome-related project? All right, okay, I can work with that. So for everyone with your hands up, thank you for your service. Everyone with your hands down, I hope that we can become best friends on this magical journey I'm about to take you on. So despite what your answer was, I think we can agree that microbiomes are essential for plant and animal species. The microorganisms that they contain provide essential services, they produce many compounds, and they can directly impact things like the host phenotype and fitness. And so as a result, there's a growing appreciation for the need to study microbiomes and to characterize the biodiversity that they contain. So one great way to do this is through metagenomics. And here the goal is to characterize the complete genomic content for all the microbial species present in a microbiome sample. And so in this cartoon example, let's imagine we have a microbiome. Those circles represent all these different bacteria and archaeal genomes. We do bulk DNA extraction. We follow this up with sequencing, shotgun sequencing, and then we perform metagenome assembly. And at the end of this process, what we ideally want is one representative genome for each of the species present in the original microbiome sample. And we call these genomes MAGs, and I'm going to keep saying that throughout the talk. And MAGs refers to metagenome assembled genomes. And this differentiates it from other types of technology that we can use to produce genomes. So for example, isolate sequencing. And I should mention that metagenomics is also very distinct from metabarcoding. And in that case, we're really amplifying like a small portion of the genome, like 16S or ITS, to identify particular species. But with metagenomics, we have complete access to all the species in the sample, and conceptually, in principle, the complete genomic contents as well. So it seems like a relatively simple idea, but in practice, metagenome assembly is actually very, very difficult. So there are quite a few challenges here. The first is that there are multiple species present. Pretty much every other presentation we just heard was focused on targeted sequencing of one organism. We violate that assumption when we're doing metagenomics. The second challenge is that these species are present in uneven and unknown coverages. And then the third challenge is that there's lots of repeat regions and intergenomic overlaps, not only across different species, but sometimes also within the same species that occurs in the form of strain level variation. And so I would actually argue that metagenome assembly is the most difficult assembly issue out there still. And I know that that's a big statement in this crowd, But I really do believe that, and it's a tremendous challenge. And so historically, metagenomics has been studied using short-read technology. And so I want to run through an example of what that looks like. So again, we do our bulk DNA extraction, we do our sequencing, we end up with millions of short reads. After assembly, what we typically end up with is lots and lots and lots of very short to medium-sized contigs. And so it is incredibly rare to assemble a complete genome for any one of the species present in our original sample. And so this contig set really represents a lot of different fragmented genomes, and we need to sort of make sense of that. And so we use what we call binning methods. And so the goal of binning is to identify contigs that belong to the same putative species and group them together to sort of make the genome whole. And when we apply these binning approaches to these short read assemblies, what we end up with at the end of the day is really what I would describe as draft quality mags. So each of these mags is composed of dozens to potentially hundreds of contigs. And even though maybe the core set of genes that we expect is there might be large parts of the accessory genome that are totally missing, and we have no way to measure that. And every time we introduce a new contig to a contig set for a mag, we have the potential to introduce error and do cross-species contamination and things like that. So this is less than ideal for a lot of reasons, especially if we're using these as resources and databases to do other studies on top of. Now with hi-fi sequencing, the first big point that I really want to emphasize is that the reads that we get off of the sequencer are often the same size or larger than the contigs that we can assemble with short reads. So we're starting this assembly process at a fundamentally different point. And because of that, it's routine that we can actually assemble complete genomes for some of the species that are present in really high abundances. So those circles that you see represent complete closed bacterial genomes. That's possible now. And in addition, we get some fragmentation, but the contigs that we generally produce are much, much larger in size, so hundreds of thousands of kb to potentially millions of bases. And so binning is a lot easier when you have these longer reads. It's a much simpler problem. But we still need to use binning approaches, even with these long read assemblies, to maximize the number of mags that we're obtaining at the end of the day. But the end result is what I would describe as really high quality mags, and those ones that we have that are complete circular contigs with high coverage, those I would argue are reference quality. So really superior in a lot of ways. And so long reads have really, as they've changed the game for targeted genome sequencing projects, they're really changing things for metagenomics as well. And early on in 2022, we published a paper describing a new assembly algorithm for metagenomics that's specific to hi-fi reads. So this is called hi-fi ASM meta. And importantly in that publication, we showed that hi-fi ASM meta produces more total mags, and even more importantly, more of those single circular contigs than any other algorithm out there currently. And so this represents a tremendous leap forward in terms of the tool set that we have available for doing this kind of assembly with hi-fi reads. And here I'm showing you an example of what an assembly graph looks like. So this came from a human gut microbiome sample. You can ignore the nightmare balls of yarn in the top left corner. Those are just sort of typical of metagenome assembly. It's where a lot of contigs can potentially be joined together. Illustrates that point that I was making about a lot of shared overlaps. But what I really want you to focus on is the first 4 or so rows, and you may see that there are some circles there. And in fact, there are 56 circular contigs present in this assembly graph. And they range in size from 1.5 megabases up to about 6 megabases. And just to put this into context, a couple of years ago, getting 1 circular contig in an assembly graph was tremendous. And the fact that we can get 56 from 1 sample on screen right now it's truly incredible. So this is, long reads are really kind of taking things leaps and bounds ahead. But you may also notice there's lots of other stuff there too, right? So we have plenty of linear contigs, which again represent these fragmented genomes. And this is again why we use binning approaches, so we can maximize the number of mags that we get from this contig set. So as part of my job at PacBio, I evaluate a lot of tools. I develop new tools, I develop new workflows, and I tailor these specifically to long reads. And I do a lot of work with metagenomics. And so I've produced a tool set, really like a workhorse tool set that you can use for doing metagenomic analyses with hi-fi data sets. And so all of these tools are open source. They're freely available on GitHub under Pacific Biosciences. There's a PB Metagenomics tools repo, I encourage you to check it out. And so in this case, I'm showing an example of a workflow that's used after we do our assembly. So it makes sense of these contigs by performing these binning steps, doing QC, and then doing taxonomic identification at the end. One of the things I want to point out, and I'm sure many of you are aware of, is that a lot of the tools that we use for short reads, they don't work very well for long reads, right? And so there's some weird idiosyncratic things that happen when we have really high quality long read metagenomic assemblies. So some of these binning tools will actually throw away complete circular contigs, which is bad because we want those genomes. And so with the tool set that I developed, I benchmarked that against the standard binning pipeline, and I found that the HiFi specific one results in a 15 to 64% increase in total mags, and more importantly, an 8 to 100% increase in those single contig circular mags. And so the thing that I really want to emphasize is we have tools available to make these analyses easier. And if you're not using these tools, just to be aware that you need to use a tool set that is tailored to your particular data type. Because there are issues with long reads that don't occur with short reads, and we need to make sure that we're aware of those. So in many ways, 2022 was really a breakout year for HiFi metagenomics, and it kicked off with this really great paper led by Derek Bickhart and Tim Smith that was focused on a deep sequencing study of a sheep gut microbiome sample. And they really showed the potential of using long reads for metagenome assembly. And this was followed up by 14 or so other additional papers that kind of showcased different aspects of metagenomic analysis. And in a lot of ways, these studies have sort of laid the roadmap for how to do analysis for metagenomics with HiFi reads. And one of the other reasons I bring this up is because along the way, these studies have produced associated data sets. So there's now a wealth of HiFi metagenomics data sets out there that we can use for a variety of purposes. And so here I'm trying to show a summary of what those different data sets look like. So on the figure on the left, you can see on the x-axis is the HiFi read length. That's the average for the data set. And then on the y-axis, you can see the total number of HiFi reads. What's really cool here is that we have lots of diversity in terms of sample types. So we have human gut microbiome, we have animal gut microbiomes, and we have some amazing environmental samples. So we have hot spring sediment, we have activated sludge, we have photobioreactors, and we have seawater. So a diversity of different sample types, you can kind of see what that might look like if you wanted to sequence this yourself. And the different colors represent different multiplex strategies. And so you can also kind of get a sense for how much data you can generate using different experimental designs. And the last thing I want to say is related to data accuracy. So the figure on the right shows the accuracy of the reads. And Q20 is a number that's thrown around a lot, and that represents 99% accuracy. You can see that most of these data sets actually fall between Q30 and Q40, representing 99.9 to 99.99% accuracy. And it's really this high accuracy that makes these data sets easy to work with. It makes assembly so much easier when we don't have to do a lot of error correction. And it makes taxonomic profiling and all these other things like gene discovery so much more simpler when we don't have to worry about these errors. So for the purposes of this talk, I wanted to just grab sort of a subset of these data sets and perform an assembly using HiFi ASM Meta, do the evaluation with the HiFi Meg pipeline, and just kind of show you what those results would look like. So I'll start out by zooming in on one sample in particular. So this is the sheep gut microbiome from the paper that I mentioned previously. So again, I reanalyzed these data. This is not directly from the paper. And after the assembly and the evaluation, I recovered 285 high-quality mags. And so the figure on the left is showing some of the characteristics of these mags. So on the x-axis, we have the completeness of the genome, and on the y-axis, we have the potential contamination present in the genome. And each dot represents an individual mag, and they're color-coded by the number of contigs they contain. So yellow is a single contig and generally circular. Anything that's not yellow is something that I recovered that has 2 or more contigs through that binning step. And what we really want to see is a bunch of yellow dots in that bottom right-hand corner that's outlined. So this represents a really incredible zone of high completeness above 95% and very low contamination below 5%. And if we look in that zone, there's 140 mags there. And more incredibly, 94 of those represent those single circular contigs. And I would describe those again as reference quality genomes. Those came straight from the assembler itself, which is, it's really remarkable. And the figure on the right is showing sort of the distribution of genome sizes and average depth of coverage. And the thing that I want to emphasize is that we can assemble genomes from 1 megabase up to 6 megabases with no problem, with anywhere from 5x coverage to over 800x coverage. And here, sequencing depth is really kind of related to how abundant that species is in the sample. So things that have high abundance, we're going to assemble those first. And when we do deep sequencing, of course, they're going to have much higher depth of coverage. So that was just one sample. Let's zoom out. I analyzed 12 data sets in total. And the figure is sort of summarizing the total number of MAGs that I recovered. So the number at the top of the stack bar plots represent the total number of MAGs. Dark purple represents those MAGs that are represented by a single circular contig. And then light purple is showing MAGs that have 2 or more contigs. So you can see those relative proportions. So across the samples, I recovered anywhere from 39 to 285 high-quality MAGs. If we aggregate that across these 12 data sets, that's over 1,200 total MAGs. And 450 of those, again, are these really high-quality, single contig circular MAGs. And when I did the taxonomic assignment, I was able to assign around 75% of these genomes to a known species. So it's 460 species in total, about 1,000 of those MAGs. So we do have multiple representatives of some of these different species, which could also represent strain level variation. But interestingly, 25% of those had no close match in any of the genome databases. So these are likely to represent new biodiversity. And if you think about those numbers, that's almost 300 MAGs that I did. This was an analysis that I ran in 3 days. If you scale this out across many samples and different sample types, you could imagine the scale of biodiversity discovery that this allows, which is, to me, it's really encouraging. And the last thing I want to say here is that you'll notice that there's quite a bit of variation in terms of the total number of MAGs. And so what this really comes down to is how much diversity is in the sample and how much sequencing depth did we apply. So some samples had more diversity than others, and some of these samples had higher depth of sequencing. So a common question that I get is, how much sequencing would I need to do to get good results for metagenome assembly? And I took a look at that in pretty close detail recently. So we did deep sequencing on a human gut reference standard that's produced by Zymo. And we sequenced 4 SMRT cells in our Sequel IIe system that generated about 90 gigabases of data. I then took that full data set and downsampled it to represent 3 cells, 2 cells, 1 cell, 2-plex, 4-plex, 8-plex, all the way down. And for each of those data sets, I ran the assembly and the evaluation, and the numbers you're seeing here are the total MAGs that came out the other side. And so you can see there's a pretty predictable relationship here. This is a log relationship between sequencing depth and the number of MAGs that we get. And so we generally recommend starting out with 1 SMRT cell to be safe because we don't know what's in our sample a lot of the time. That gives, it's pretty much guaranteed to give good results for most of the samples. And then you can kind of adjust that based on how that first sequencing run goes. But we realize that for a lot of projects that want to do scale, this may not be the most cost-effective solution. And the good news for us is with the Revio system, we now expect a lot more throughput. And so more exciting for me than anything else, we just got some metagenomics data sets finished running last night. And so here I'm showing the results of that. So we sequenced that same Zymo fecal reference. We ran 2 replicates on Revio, and the HiFi yields look great. The results are really consistent between both replicates. So we got about 68 gigabases of data. That translates into 8.7 to 9 million reads, an average read length of 7.7 kb, which is pretty good for metagenomics because the DNA extractions are harsh and we tend to be pretty brutal on the DNA. But somewhat surprisingly, but also amazing, is the median QV score jumped up quite a bit too. So instead of seeing the range of 30, 35, the Q score here is actually 46. And so this shows some of the power of using something like Google Deep Consensus on Revio for calling HiFi reads. So this is all very encouraging, and then as soon as the night is over, I'm going to kick off some analyses to see how many MAGs we have in here. But to kind of put this back into context of what we were just describing, so with 3 SMRT cells, we can get about 164 MAGs, and that's on the Sequel IIe. That is what I would consider sort of the deep sequencing project level. But now if you look, that's the equivalent of 1 SMRT cell on Revio. So we're taking deep sequencing and making that kind of routine. And the 1 SMRT cell on Sequel IIe, that now represents a 3-plex on Revio. So you can increase your sample size if you want to by multiplexing. And then I did just a quick cost comparison. So I added up the total amount of cost for sequencing 3 SMRT cells on Sequel IIe. I divided that by the number of MAGs. and that totaled to about $24 per mag. And when we do the same calculation for Revio, that drops to $6 per mag. And so not only are we getting higher throughput and increasing mag recovery, we're also doing this at a severely decreased cost. And so I expect that this is going to enable a lot of new metagenomics projects to kick off, especially those that are interested in sequencing at scale. So to summarize, long-read sequencing has been truly transformative for metagenome assembly. We can obtain high-quality genomes for non-culturable species. This is simply just not possible using isolate sequencing or short-read sequencing. And this allows us to discover and characterize new microbial diversity to get these high-quality genomes that we can then apply to these reference catalogs and use for other downstream analyses. The Revio system, as I said, will sort of allow us to do these deep sequencing level projects as sort of fairly routine with one SMRT Cell. And again, all of the development work that I do is open source and available on GitHub. And all those data sets that I show, you can find links to where to get those and all the metadata for those also on GitHub. And so if you're interested in long-read metagenomics, I would encourage you to contact us directly. I'm leaving my info down below. With that, thanks for your time, and yeah, thanks for being here.